Life science researchers today are intensely interested in the mechanisms of disease and toxicity. They need to know what is happening at the biological level to understand, for example, why one person gets sick and another doesn't, why a particular chemical agent or drug is toxic to one person but not another, or why one person responds to a particular treatment and another doesn't. Genomics, transcriptomics, proteomics and metabolomics all offer clues, but to really understand disease mechanisms and toxicity pathways, no single discipline is enough. That is why we at Agilent are taking an integrated approach, looking at biology as a whole system. We are supporting researchers with tools to combine and correlate data from multiple omics to deliver that deep understanding of mechanisms and pathways. Additionally, through our Life Sciences Center in Bangalore, India, Agilent is collaborating with some of the world's leading researchers who are engaged in using this approach to system biology, biomedical and translational research. Those collaborations ensure that we really understand their needs and we deliver the solutions they need to be successful.
Narayana Nethralaya, one of LSCI's collaborators, is a super specialty eye hospital in Bangalore.
Hi, Dr. Mallipatna. I heard you are working with eye cancer in little children. Can you tell me a little bit more about your work?
Sure, Dr. Ghosh. I'm very interested in a cancer called retinoblastoma, which is a cancer that affects the eyes of children under the age of 5. It's usually detected by a white reflex in the eye, but in India, unfortunately, sometimes we detect it too late. We're hearing things about a biomarker discovery that is new, which can probably even discover the cancer earlier than what we're doing now.
Certainly. I think especially for something like this, an integrated biology approach would be the most appropriate, where you take a holistic approach and have genomics, transcriptomics, proteomics, metabolomics all integrated, and the data collated is brought together by a bioinformatics consortium which measures and analyzes everything in holistic form and gives you very specific biomarkers that can be related to progression of the disease. It is something that requires a lot of niche skills and niche, highly sophisticated infrastructure.
Do you know any places that do this kind of work?
Agilent Technologies does a lot of that because they build a lot of these machines and they build a lot of the software also. So, shall we go visit Agilent?
Sure, let's do that.
Hello, Sudha.
Hello, Arka. How are you? Welcome to Agilent.
So, I'll introduce you to our friend, my colleague Dr. Ashwin Mallipatna and Dr. Sudha Rajgopalan. So, could you tell us more about the integrated biology approach at Agilent Technologies?
Sure. Let's go inside and I'll introduce you to the team and we can also see the lab as well. We at Life Science Center develop applications in different areas: pharma, biopharma, metabolomics, proteomics, genomics. And we also have several collaborations both inside the country as well as outside the country. There are several integrated biology projects going on. Let me introduce you to the team of scientists who worked on the rapamycin study. Neelanjan will explain that study a little bit in detail.
As a proof of principle, we used a multi-omics approach to study the effect of immunosuppressive drug rapamycin on human embryonic kidney cell line, HEK293. We used an integrative approach by using the Agilent best-in-class instrumentation, software tools, and the sample preparation kits. Using our GeneSpring integrated biology software, we showed that rapamycin affects a lot of biological pathways like protein translation, DNA repair, purine metabolism, and many of these pathways has been corroborated with other studies as well. Here we see integrated approach where combination of genomics and metabolomics shows us some of the key pathways affected by rapamycin. We also have automation to help in our high throughput studies. For example, we are currently collaborating with Baylor College of Medicine on a prostate cancer health disparity story. Here you are analyzing 200 samples and we'll be doing the transcriptomics here and the metabolomics will be done at their end. And at the end of the study, we are going to bring in 2 data and analyze them using our GeneSpring integrated biology software. We are currently working on a metabolomics project involving medicinal plant. Salman is going to explain more about this project.
Agilent is a leader in metabolomics area. It has all the instruments and software necessary to perform comprehensive metabolomics analysis. We are currently working on medicinal plant project and we are looking at all its metabolomites using LCMS, GCMS as well as NMR technologies. In fact, We can take the metabolomics data, combine it with other omics data such as proteomics, gene expression, using our GeneSpring software.
Oh, so that's fantastic. So actually for the retinoblastoma samples, we can look at the metabolomic profile of that, including the gene expression as well in the same sample.
Absolutely.
And then follow that up with a more targeted proteomic approach.
Absolutely. In proteomics, you can also do phosphoproteomics as well.
Phosphoproteomics is a branch of proteomics where we try to identify, catalog and characterize proteins with a phosphate group as a post-translation modification. Another project we are working with Karnataka University where we are trying to understand the effect of fungal lectin on cancer cells using our own phosphochip-based solution.
Thank you very much for showing us the Life Sciences Center. I'm actually very impressed with all the facilities and the expertise that's available here. I think with this we should really be taking the retinoblastoma project forward.
Thanks for visiting us. We are looking forward to working with you.
It's been a pleasure.
Thank you, Sudha.